first of all, thanks so much for inviting me. It's a real pleasure and an honor to be here. I live in Maine, but I'm from Colorado, so it's always fun to come back west of the Mississippi whenever I can get a chance. So I hope you'll indulge me starting off with some Acadia National Park eye candy here. It's just prettier than a title slide, I think. I'm a staff scientist and a member of faculty at the Mount Desert Island Biological Laboratory. Geographically, Acadia National Park dominates Mount Desert Island. So it's not much of an exaggeration to say this is my backyard. I'm very happy about it. I wanted to share that. The laboratory that I work at, though, is just outside of the park. So there's Maine there. Acadia National Park is in down east Maine, and Mount Desert Island is in that blow-up map there. And the green parts, I don't know if you can see from where you're sitting, are the park, and the arrow is pointing to the location of the laboratory. The reason I have that there is because I want to dispel a couple of misconceptions about where I'm from before I get started on my Charles Darwin stuff. One common misconception is that the MDI Biological Laboratory is the same thing as Woods Hole, and it's not. Another misconception is that it is the same thing as the Jackson Laboratory, which is a much larger laboratory. It's a mouse genetics research institute, and it's right here in Bar Harbor, and it's also a different laboratory, so don't confuse those things. What we are, so that's what we aren't, what we are is a nonprofit research institution with a 116-year history, including some seminal work in renal physiology and environmental toxicology. Today, we tend to focus on a combination of regenerative biology and aging research, and it may seem like a strange bedfellow, environmental research, which is my area. But if you look back at the renal physiology and environmental toxicology background, you can understand how those two research foci sort of sprang out of the history of the lab. Okay, so one more piece of eye candy. Before I talk to you about Darwin and the Beagle Project, I'll give you one sentence about what I actually do for a living. I'm working with Acadia National Park and the Schoodic Research and Education Institute, which is actually located across the water over there, working on a National Science Foundation-funded project to combine DNA sequencing and citizen science to try and expand the capacity at which we can do environmental research and ecology. So Steve's talk was concerned with variation within one species, and the DNA sequencing and the DNA work that I do is concerned with variation between species, and we try to use that to tell different species apart so that we can identify them accurately when we find them in the field, and then use that information to answer big questions in ecology, like how will climate change affect the different plant and animal species around the world. So here's the title slide. But today I've been asked to talk to you about some of the things I actually did before I moved to the Mount Desert Island Biological Laboratory while I was a postdoctoral research at the Natural History Museum in London, and also as part of my role with the HMS Beagle Project. So my presentation tonight is going to be part travelogue, part science talk, and it's going to be based on the route of the voyage of the Beagle. And so I'm going to use the voyage map as a kind of guide through my talk. So first, an overview of the voyage. Actually, about 5 years ago, I looked for some good maps of the voyage of the Beagle and could not find one that I liked, so I made this one for myself, and one day I'll get around to making a really nice large format poster to be sold at events like this. But I haven't got there yet. So Charles Darwin was on the second of 3 voyages of HMS Beagle, each captained by 3 different people. The voyage captain for Darwin's voyage, of course, was Robert Fitzroy, and that voyage lasted 5 years, and Darwin was chosen to be a gentleman companion for Fitzroy. Fitzroy wanted someone to talk to, lest he suffer the fate of his predecessor, Pringle Stokes, who took his own life on the first voyage of the Beagle in the dark depths of Tierra del Fuego. Darwin was so looking forward to the voyage, but then realized very soon that he was going to be suffering from seasickness almost the entire time. And I think it's a great mark of his character that even though he was what we would call today a rich kid and could have left the voyage at any moment and gone back home on the first packet ship, he stuck with it for the entire 5 years, and he was pretty much vomiting that whole time, so props. Because of the seasickness, he spent as much time as he could off the boat, and he did a lot of trekking with his geological hammer and his pistol and his collection equipment. In fact, I think he spent about 3/4 of his time during that 5 years with his feet on land. So I want to break from that overview just briefly to remind you that at the time, even though there are beards that are going to be worn later at the after party, at the time of the voyage, Darwin was just 22 years old, which means he was about halfway in between these two pictures right here. So even though we often think of him as, and often he is depicted as, a bearded old man who has changed the world already, we should imagine him when he's on the Beagle, something like this. But back to the map. So the voyage lasted 5 years. It went all the way around the world, so it was a circumnavigation. And most of that time was actually spent in South America. And if we zoom in a little bit further to the southern portion of South America, we can see how much the Beagle actually looped all around different ports, different locations. This is because the charge of HMS Beagle was to survey the coast of South America. So they did a lot of back and forthing to go back and get points that they hadn't been able to survey previously, but they had to go back and get supplies, and so then they went back later. And often Darwin would get dropped off, and the Beagle would go swooping off to like the Falkland Islands, and then they would come back and pick him up later. And these little dotted lines on the map here are Darwin's inland voyages, where he either went on foot or by horseback. And for example, he went on horseback with the gauchos famously, and learned to live the way that they live on the Pampas. One interesting point here, you see this? So they go around Cape Horn and they get to this point, and then they have to turn back again. That's not fun, people. That's a really difficult thing to do even today. And I have this phrase here, sorely tried, because they had at least one serious brush with death on this voyage. And the Beagle almost capsized off Cape Horn. This is a painting by John Chancellor of that event as described in Fitzroy's logbooks. So I want to read just a little portion of that. Fitzroy wrote on the 13th of January, 1833, "Soon after 1, the sea had risen to a great height, and I was anxiously watching the successive waves when 3 huge rollers approached, whose size and steepness at once told me that our sea-boat, good as she was, would be sorely tried. Having steerage way, the vessel met and rose over the first, unharmed, but of course her way was checked. The second deadened her way completely, throwing her off the wind, and the third great sea, taking her right abeam, turned her so far over that all the lee bulwark from the cat head to the stern davit was 2 or 3 feet underwater." So we almost lost Darwin and all the crew of the Beagle right at that moment, and then it would have been Wallace on the Palouse that we would be celebrating today. Darwin also lost a number of the plant specimens he had collected in Brazil in particular in this near tragedy. So after all that time in South America, Galapagos was kind of an afterthought. We kind of like to imagine, or the way the story is told, the purpose of the Beagle Voyage was to take a young Charles Darwin around the world, especially to the Galapagos, where he discovered evolution by natural selection. But mostly it was to survey South America, and he was Fitzroy's gentleman companion and was just getting seasick the whole time and collecting lots of specimens. And then they go to Galapagos to get some water and some tortoise meat because, hey, they're on their way home and they're just going to stop for a couple weeks. They were only on Galapagos for 3 weeks, and it's interesting, after they left there and they come around to Australia, you'll notice that they don't go up to the northern tip of Australia where the town of Darwin is located up here. That's because Darwin never visited that town that bears his name. It was named by his crewmates, who on the third voyage went there and named it after their friend, Charles Darwin. So now back to the big picture of my talk. That was the voyage route, a little bit about the voyage, a couple of highlights, or a low light, whatever you call that sorely tried picture. I'm going to tell you today about some of the projects and events that I've been fortunate enough to be involved in that have to do with different points on the voyage. From London to Parachi in Brazil, which is just near Rio de Janeiro, southern Patagonia and Tierra del Fuego, Santiago, Valparaiso, Galapagos, and I don't think I'll have time to talk about South Australia today, and then of course London at the beginning and the end. So like the voyage, we'll begin and finish in London or in the UK. So as I said, I was a postdoc at the Natural History Museum in London from 2003 to 2010, where I worked on a whole range of molecular systematics projects. But also from 2007 to 2009, I was seconded to be a coordinator for the science aspects of Darwin's 200th birthday program celebrations. This was a series of events, hundreds, thousands even of events around the UK and around the world celebrating Darwin's 200th birthday, and the Natural History Museum was sort of the epicenter of this big, giant birthday party. And some of you will know this, but maybe not all, so I'll say it. We were really celebrating not just Darwin's 200th birthday, but in 2008, we were celebrating the 150th anniversary of the publication of the reading of Darwin and Wallace's paper at the Linnaean Society announcing the discovery of natural selection. And then in November 2009, the 150th anniversary of the publication of On the Origin of Species, which is for sale just outside. How'd I do? So really this whole period, this whole Darwin anniversary period as a biologist, as a human, was just, I was a kid in a candy store. This was so fun for me. Even though I was having to take some time away from my research, I wouldn't go back and change that. There was, of course, a big birthday party, and here's Darwin's 200th birthday cake with Diplodocus dinosaur skeleton for scale. Actually, the museum just announced they're replacing Dippy the dinosaur, which some of you may have seen, I don't know, with a blue whale in the central hall. And I've been told we're going to have a birthday cake later at the after party, so I suspect it's not dino poo sized because this is the tail of the Diplodocus is right here, so it looks like it's just been dropped down. A very bad place to put the cake. So hopefully our cake tonight is not dino poo sized, but maybe that's a good thing. There were special exhibitions at the museum. This one, the Darwin Exhibition, also came to the American Museum of Natural History. Some of you may have seen it there. And there were conferences in places like Istanbul and, of course, Cambridge. Here's a stained glass of Darwin in Cambridge. And at the Natural History Museum, I worked on a whole range of projects connected in some way or another with Darwin's legacy. Now, a museum is first and foremost a specimen repository. And so one big question we had was, well, How many specimens does the Natural History Museum hold that were collected by Charles Darwin? Now, you'd think that we would know that and that every single one would be in some kind of gilded case right in the front entrance, or at least I would think that. But actually, in a place with 70 million specimens, it's really hard to actually know how many you have collected by a single collector, even one as famous as Darwin. So we did a survey where we interviewed curators, especially the older curators were an amazing trove of information. And we thought we'd be lucky if we found that there were a few thousand specimens in the museum collected by Darwin. We found actually there were well over 10,000 specimens. So here's just a few little pictures of some. There were wonderful fossils, including this skull of a Falkland Islands fox, skeletons of pigeons that the fancy pigeons that he later bred, and with his own notes directly on the bone here. Lizards, corals from the Cocos-Keeling Islands to the northwest of Australia, and of course beetles. Darwin loved beetles. Mollusks, a parrot fish. This is the parrot fish, by the way. Not this. This is Ollie Crimmen, the curator of fishes. I think this is the octopus that he found. This was like the first specimen he collected on the voyage of the Beagle at Cape Verde. So that's an exciting specimen. Obviously, these are not in any kind of taxonomic order. So also during this time, when I was in London, I became involved with the HMS Beagle Project. We are a UK charity that are fundraising to sail a new Beagle around the world in the wake of the second voyage of the Beagle, the one that Darwin went on, but carrying scientists, communicators, teachers, students, and members of the public as kind of a floating science festival. Darwin said, "The voyage of the Beagle has been by far the most important event in my life, and it has determined my whole career." So if an adventure aboard a ship like the Beagle for a 22-year-old English boy, really, he was a boy, I guess, at that point, could have such a great influence on him, maybe such a similar voyage could have an influence on people today. And so we hope the new Beagle will really be a flagship for science today. Now, we originally focused on raising money to rebuild the Beagle that would look like a replica on the outside, but would be a slightly more modern inside. We actually, over the last couple of years, have shifted towards finding a partner vessel that is about the right size to be the Beagle, and make some changes to it and use that. And I'm delighted to be able to tell you that we've had some very significant fundraising progress over the last couple of months that I'm not allowed to talk about. But the project to obtain a ship is looking pretty near at hand. So hopefully the new Beagle coming soon to a port, probably not near you, but you can follow the Beagle Project on hmsbeagleproject.org or on Twitter at Beagle Project or on Facebook. So with the Beagle Project, I've been fortunate to travel to some of the Beagle Voyage sites to spread awareness about the project and establish some new sister organizations, including La Fundación Beagle. You pronounced that absolutely right. We went to Brazil first to work with a tall ship, the Tocorime, which translates roughly to Spirit of Adventure in Portuguese. This was a lovely tie-in to HMS Beagle, as the original HMS Beagle actually did have a sister ship named the Adventure. So this is one of Conrad Martin's paintings from the Voyage of the Beagle, and see how there's two ships. This is the Beagle here, and this is a smaller ship that Fitzroy purchased to help with the surveying called HMS Adventure. So we held a week-long series of events in Brazil. We had educational and media events and a science symposium, and these are a bunch of local school kids aboard the Tocorime. And we timed the workshop to coincide with direct overhead passes of the International Space Station, and we're collaborating with NASA, I'll get to this in a minute, so that we could practice some of our methods for correlating imagery and data collection during the voyage of the new Beagle someday. So we set up a live Q&A between the local school children and our collaborating astronaut, Mike Barrett, who was aboard the space station at the time. And this is one of the best experiences of my professional life, because these kids learned their questions in English for the astronaut, and they were so nervous they had them all wadded up in sweaty little balls, and then when their time to ask their question, look how earnest she is. They asked their question, and then they said, "Over." And then they waited for the answer, and then Mike's voice would come over the ham radio and answer their question, and they were just, so delighted. Their parents were there crying, and it was wonderful. In Chile, then we visited Santiago and Valparaiso for a range of meetings with stakeholders from the government, from science and industry, all looking for fundraising opportunities. And we also visited Tierra del Fuego. We got to fly on a little Navy plane to get down there. It was kind of exciting. Where we got to visit this marker of one of Beagle's anchorages along the Magellan Strait. This, by the way, is another co-founder of the Beagle Project, David Lort Phillips. And this is Randall Keynes, Charles Darwin's great-great-grandson. We also visited the location of the burial of the Beagle's first captain, Pringle Stokes, who I told you about earlier, had committed suicide there. And this is an amazing place to visit. It's stark landscape, it's cold, it's rainy, it's just kind of bleak. There aren't many trees, and on this hillside are just this little tiny patch of white crosses, and the big one is for Pringle Stokes, and Randall is here. It sort of looks like he's wiping away a tear. I like to think he's wiping away a tear. He's probably just, rubbing his face or something, but I think it's a very evocative photo. Most excitingly for me on this trip, I finally got to lay eyes on the famous Beagle Channel, named after the Beagle, you might have guessed. And the relationships we established in Chile on this visit are what led to the founding of the new sister charity, La Fundación Beagle, and I'm going to be working soon on establishing a third sister charity to be based in the United States. So watch this space. Now the moment you've all been waiting for, Galapagos. Another project I got to work on during this Darwin 200 period was I got to extract historical DNA from these specimens collected by Charles Darwin and Robert Fitzroy themselves in Galapagos. Now, this wasn't just for fun because it's a cool historical project. This was part of a conservation genetics effort. So it was these mockingbirds. These are not finches, these are mockingbirds. It was Galapagos mockingbirds, not Galapagos finches, though they're more famous, that if you could say, which you really can't, that Darwin had any kind of eureka moment during the voyage of the Beagle, it had to do with these mockingbirds, not the finches. Not the finches. So we know this because, first of all, he didn't recognize until much later, after Gould looked at his finch specimens and told him that there were separate species on all the different islands. He just thought they were all little brown birds. The mockingbirds, on the other hand, he recognized immediately there were different species on different islands. And he wrote, and I know you can all read Darwin's handwriting, not really, so I'll read it to you. He wrote, "When I see these islands in sight of each other and possessed of but a scanty stock of animals tenanted by these birds, but slightly differing in structure and filling the same place in nature, I must suspect they are only varieties. The only fact of a similar kind of which I am aware is the constant asserted difference between the wolf-like fox of East and West Falkland Islands." Remember the Falkland Island fox skull he collected? "If there is the slightest foundation for these remarks, the zoology of archipelagos will be well worth examining. And this is the important part that's highlighted in yellow here. For such facts would undermine the stability of species. So he's still on the voyage of the beagle at this point, and he's recognizing that the birds, these mockingbirds, and the foxes, and also the tortoises that he sees on Galapagos, are evidence that species change. But today, this mockingbird is one of the rarest species in the world. It's actually extinct on the island of Floriana, the Floriana mockingbird, where Darwin and Fitzroy first collected it. And it's hanging on two little islets that are too small to even see in this satellite image just off of Floriana, where the invasive animals that live on Floriana that humans have brought have not been able to kill the birds or destroy their habitat. In recent years, these invasive species have been largely removed from Floriana through culling and other measures, including rat poison. It doesn't sound very appealing, but this is what you have to do when you bring rats someplace and then want to get rid of them, which had destroyed the native Opuntia cactus forests. As you can see, this bird likes to be on this cactus. That's because the cactus is its food plant. So when things like feral pigs and goats destroy the cactus, the birds don't have anything to eat. So now that conservation biologists have removed a lot of these invasive species from Floriana, they would like to be able to reintroduce the mockingbird from those two little islets back onto the main island. But then there was a question, what about the genetic diversity of the birds after they've been surviving on these two little islands for so long? How does that relate to the former diversity of the birds on the main Floriana island, and how can we recreate as best we can the population that was there? The project was led by a group of researchers from Zurich, Paquita Hoch and her advisor Lucas Keller, and I helped them to gain access to the only specimens on Earth that we know about from Floriana that were collected on Floriana by Darwin and Fitzroy. And these are some of the mockingbird specimens in the collection of the Natural History Museum. And what we did is we took tiny little pieces of tissue from the foot pads of the mockingbirds. You can see the little triangular shape that's been taken out here. So we succeeded in getting samples, and Paquita and I independently extracted DNA in Zurich and London to make a replicate of our study as a precaution against contamination. They were especially worried their lab in Zurich, where they work with a lot of mockingbird specimens, that they could get contaminating material from that lab. In my lab in London, we'd only been working on plants, so there was a very low likelihood we would get mockingbird DNA in there. So all of this is cutting a very long story short, but all of this resulted in the publication of Saving Darwin's Muse, a paper that we published in the Royal Society's Biology Letters Journal, and it was extremely exciting and gratifying to learn then that our recommendations were taken up by the conservation biologists working in Galapagos, and they decided to introduce birds taken from both of the satellite islets instead of one or the other. Those were the original options that they had based on our recommendations in this paper. So there I was. I'd worked on all these Galapagos birds. I loved the whole project, everything about it, but I had never been to Galapagos myself. And in 2010, my chance arrived, and I received an email about an opportunity to accompany a group of high school students, all girls as it so happens, who had won a Darwin-related science video competition hosted by the Wellcome Trust, which is sort of the UK's version of the Howard Hughes Medical Institute. And I was sort of a scientist guide for these girls. When we got to the islands, I think probably 2 minutes after we got off the airplane, the first finches. Wow. But you know what I was there for, right? Not the finches. I didn't have to wait long, so Galapagos mockingbirds and most birds there are very tame, and they'll hop up on your shoe and untie your shoelaces. They're incredible. Now this This is not a Floriana mockingbird. There are no Floriana mockingbirds on Floriana, as I was just describing. This is one of the other species of mockingbirds on Santa Cruz. But they all look pretty similar, but not identical, which Darwin noticed because they're different species. But again, they're so tame. There's no zoom lens here. This is just an iPhone picture going like this. We also saw the iconic giant tortoises and marine iguanas. This was just heaven to me. We visited a volcanic caldera on the island of Isabella. And we took, most exciting of all, we took a day trip to the Beagle's first landing site in Galapagos. So you can walk along this path on San Cristobal and visit the cove where the Beagle first landed, and Darwin was rowed ashore onto Galapagos and got out of the boat and stood there. And he looked at some cuttlefish that were in the water, and he started collecting, and he probably saw his first mockingbirds there. But it was very exciting to arrive there, and then, of course, to imagine someday on the new Beagle sailing into that very spot. So I just got a few little goosebumps. So coming back to the end of our voyage, back to the UK, I was also part of a Natural History Museum project to resurvey a meadow at Down House, Darwin's home in Kent, where Darwin conducted what we think is probably among the first of any biodiversity survey ever conducted in 1855. So I don't know if you know this, but I didn't before moving over there. London is big. This red area is London, and Down House is in Greater London. You can, if you're ever traveling through, it's worth taking a trip down to Down House. You don't have to go way out into the countryside. It's just right there in London. Anyways, so some of you may have visited there. I don't know if you're lucky enough to have done that, but this is the Google map image. Down House is right here, and there's this meadow near Down House, and actually Darwin's famous thinking path comes down here and then loops around through these trees. But this meadow that's right next to it is the meadow where he conducted this biodiversity survey. So in June 1855, Charles Darwin wrote to Joseph Hooker that he intended to survey this meadow. And he was so excited, he wrote, "Hurrah, hurrah," because he had identified his first grass. And actually, his co-researcher in this project was Miss Thorley, his children's governess. And I have a feeling there were days when she did all the work, because he would have these episodes of gastric distress and sickness that lasted well beyond his voyage on the Beagle, and he would be incapacitated sometimes for days. So she was probably out there collecting and identifying a lot of the plants. That was just my little Women in Science booster. And by the end of that month, they'd identified 28 species of grass. By the end of July, they were up to 35 species, and they were still conducting the survey in August when he wrote to John Henslow, the one who had gotten him his berth on the Beagle when he was 22, that he was, quote, puzzled by an umbellifer, end quote. So here, he's unwell. It's 1855, decades after coming back from the voyage of the Beagle, and he's still just intensely curious and decides to discover every plant in this one meadow. And so I think it's just a testament to his lifelong love of exploration, really. He wanted to do this because he wanted to learn British plants, and the best way to do it is by actually getting out there and looking at them and trying to identify them. He also wanted to show the degree of diversity on even just a little small plot. And this narrative was part of Darwin's this narrative appeared in the second part of Darwin's original Big Species book, which is what he was working on when he famously got Alfred Russel Wallace's letter that spurred him to write an abstract, which became Origin of Species. But halfway through writing, so it was halfway through writing this book that he got the letter, and as he was writing the Origin of Species, he actually changed a section in the Origin to being from this meadow to being about a separate 3-by-4-foot patch of lawn near his house. But the Great Pucklands Meadow, the name of the meadow is Great Pucklands, survey is still in the Big Species Book manuscripts. So what were we doing there in Darwin's Meadow all those years later? We were interested in using the meadow like Darwin did to teach ourselves botany. But in our case, it was not just to match names to plants, but to practice a new technique for DNA-based species identification called DNA barcoding. So during the summers of 2005, 2006, and 2007, we resurveyed the meadow. And we wanted to document any change that might have taken place since Darwin did his survey and evaluate these proposed DNA barcodes, these sequences that could be used for telling different species apart in plants. So we found 161 species in 2005 and 2006 and recollected 141 in 2007, which isn't too different from the number 142 that Darwin and Thorley collected in 1855. Unfortunately, without his actual list of species, we can't know if it's the exact same species that are there or if it may just be we have the same number, but some of them could be different. The next big thing for this project is to find, at long last, his list of species, which must exist somewhere because he refers to it in other places. And there are boxes and boxes of Darwin manuscripts at the American Museum of Natural History, and David Cohn and his team there in the Darwin Manuscripts Project are going through them all the time, and I keep waiting for the day that he's going to call me up and say, I found it. So, someday. This resulted in another paper that we published in the Proceedings of the National Academy of Sciences, and I'm like the 14th author in there, that list of initials. Big science, I guess. You get lost in initials. More recently, the full story, not just the DNA data, was published as a book chapter in Darwin Inspired Learning, which is published by Sense Publishers in the UK. So that's the end of the story following the Beagle's voyage on Earth. But there's a little extension that I just want to finish with that doesn't really fit on this map. So one of the more exciting bits of publicity in the early days of the Beagle Project was this little blurb about our project in Science magazine. If you're a scientist or you know any scientists, you know that getting something published in Science or Nature, where's Steve Olson? Yes, or Nature, very, very well done, is a big deal. And so even a little news-infused blurb, I was pretty excited. Well, it turns out Science magazines are left on coffee tables all over the world. And one of the coffee tables it was left on was at Johnson Space Center in Houston. And you don't have to be able to read this. It's just proof that it happened. So at that time, we weren't actually planning our inaugural voyage, but still raising money. But nevertheless, I got an email from an email address, michaelrbarrett@nasa.gov. I'm like, oh, NASA, that's an interesting @ and it's just, astronauts these days are humble to a fault. And, you undoubtedly get all sorts of crazy emails, so count this as the latest, blah, blah, blah. I stumbled across your project. I love Darwin, I love the Beagle, I love sailing, and oh yeah, I'm gonna be in space in 2009, and your project sounds great, so let's collaborate. So I'm not an idiot. I got myself on the first airplane that I could to go to Houston and meet with him, even if it meant I had to sell a kidney or something. And I stayed at a hotel near the Johnson Space Center, and when I arrived, this is what I got, a grand tour from an astronaut. If you ever get the chance to do this, take it. So this is back during the space shuttle program. May it rest in peace. We got to see space shuttle simulators. I got to go in one and push buttons. There was the one that moves. There's Mike Barrett showing me how to push the buttons, I guess. I was doing a terrible job. We went into mission control, not the fake one they have at Kennedy Space Center, but the real mission control. the Neutral Buoyancy Lab, where astronauts practice their spacewalks, and has a full-scale, it's the largest indoor diving facility in the world, and has a full-scale space station in it. I touched moon rocks. It was pretty incredible. And we also, in addition to all this fun stuff, actually had meetings and came up with a three-pronged collaboration plan where we're going to do some public relations and outreach together, educational projects, and what we practiced in Brazil, which is to collect some samples from the new Beagle and correlate that with images taken by astronauts on the space station. So I got back to London thinking, wow, space. And I just caught the space bug again. I loved space when I was a kid, then I got sucked into biology, and then I just love space again. Michael launched on the Soyuz rocket after he launched on the space shuttle, or before he launched on the space shuttle. And it's quite an experience to watch a rocket launch where you know somebody who's on that controlled explosion, especially with some of the accidents that are burned into all of our memories. And it's just terrifying, really. So I was very happy when he got into the relative safety of orbit. So he sent down pictures to prove that he was actually in space, all weightless, and we did our event in Brazil, so this is probably likely what his position was like when he was talking to the little girl that was holding the crumpled up piece of paper. Incidentally, while I was in Brazil, I got a phone call from a strange phone number on my cell phone. If you ever get a call from this phone number, answer, because it's the space station. I tried calling it back, 0000. No, it doesn't work. But anyways, when the space station calls you, this is how it comes up on your phone. And Mike just called me and said, hey, how's it going? And I said, why does it matter how it's going down here? Tell me, what do you see out the window? What are you doing right now? What experiments are you doing? What did you eat for breakfast? How do you go to the bathroom up there? Things like that. And it was just really thrilling to be able to share that with the other people who were there in Brazil with me and the school children as well, that he was asking after them, and it was just all very exciting. So I swear this has a point. So to finish, I just want to step back and reflect a little bit about what it is about human spaceflight that connects back to biology, that I'm a biologist. What's so funny? There's adventure, there's exploration, sure, those two things are in common. But there's also a kind of self-discovery. So when the crew of Apollo 8 went to the moon to orbit around it, they saw this through their little window. And it's striking because it reminds us just how small and vulnerable we are floating on our little blue spaceship, and that we're not the center of things. It also ignited the developing environmental movement at the time that this photo was taken, and reminded us that, I guess, this not being the center of things is common to some of our greatest scientific discoveries. Think about Copernicus and Galileo. And if we think about Darwin, it's the same. So somehow our significance, our feeling of significance, my feeling of significance at least, both decreases and increases at the same time when I'm thinking about this, that we're on this little mote of dust, like Carl Sagan said, that we're just this little itty-bitty piece on the tree of life, that we're on this planet floating in space. So in this respect, there's not a very stark line between biology and space exploration. It's why I think the collaboration between NASA and the Beagle Project is so very natural. So that's it. Thank you.